Today we have chapter 2, microscope. The microscope, as you see here, it's a device. It's a device used to observe small organisms which cannot be seen by naked eye. Okay, so this is the microscope, as you see in front of you. It help us to see the living things that we cannot see by our eyes. Okay, like the things that cause disease, such as the bacteria, the viruses, some types of fungi. Okay, we go to the scientist. It was first discovered by Antoine van Leeuwenhoek when in the 7th century. This is Antoine van Leeuwenhoek. The next scientist is this one, which is Robert Hooke. He invented the new microscope. He developed the new microscope, okay, in 1665, and he observed cork cell. Cork cell is the cells of the bark of the trees. Okay, it's related to the trees. Here we have the microscope device, okay, and it's their parts. Okay, so the microscope, it has a lot of parts. We start with the first part, which is the eyepiece. This first black part is called the eyepiece, where we put our eyes to see the object through it, the eyepiece. The second part is the arm. This arm which connect this first part with the base. Okay, this arm, and help us to carry the microscope. The third part is the coarse focus knob. This one, this large button, it help us. It can be rotated to help us focus on the sample. This is the small knob, okay, which is called fine focus knob. This one help us to clarify the image, okay, so that we can see it very clear. This thing, the black part, this is considered to be the light source. It gives us light so that it will see the sample in a bright way. This is a switch which responsible for on and off, to turn on and off the light source, which is called the power switch. This part is called the base, okay, the base of the microscope. The part under this black part, okay, the square part is called the stage, where we put the sample that we are going to study. Under the stage there is a diaphragm. This one helps to control the amount of light that pass to the sample. Then we have the objective lens. This objective lens, it shows the object on or the sample which has been prepared on this stage. So it's called objective lens because it's near the object. Rotating head, this head is rotate. It helps us to rotate these lenses, okay, because they have different range of viewing the sample, or maximizing or magnifying the sample. The body tube, this help to observe the sample by using the eyepiece. Okay, so these are the parts of the microscope. Okay, the 12 parts of the light microscope. Now we have the benefits or definition of each part of the microscope, or the explanation of each part of the microscope. We start with the rotating head. This one contains mirrors inside it. Okay, This is the this is the rotating head. Okay, so we're going to explain it. It has contain mirrors and allow the body tube to rotate 360 degree. Okay, contain mirrors and help to rotate. The body tube passes light from the head to the eyepiece. Okay, this is the body tube. It helps to pass the light to the eyepiece. The next part is the eyepiece, which is the top part that we use it to see the sample. The eyepiece is where one views the enlarged objects, which means we see the enlarged object contain a lens called the ocular that further magnifies the specimen by more than multiplied by x, like we multiply it by 10x. Okay, 10x means 10 times bigger. The objective lens that we said it's near the specimen or near the sample. Objectives produce most of the magnification. The high power lens, blue stripe, you have to know the colors. If the lens contains blue stripe, it's high power, which magnifies by 40x. If it's yellow stripe, it will be low power lens and it will magnify only by 10x. If you can see this, look here, this one is blue and this one is yellow. This one is 40x, this one is 10x, this one is high power, this one is low power. We continue with the arm. The arm supports the lenses, mirrors and body tube, which means the upper part of the microscope. The microscope should be carried with one hand holding the arm and the other under the base. The sixth part is the stage. This is the black square part where it contains the sample. It holds the slide. A slide is a piece of glass, okay, that we put the sample on it and contain an opening that allow light to pass through it. Okay, it's a glass, so it would be transparent, so light has the ability to pass through it. The specimen on its way to the object. Okay, so let's make it bright. We continue with the rest of the parts of the microscope. We have the coarse focus knob. This one, it moves the stage up and down quickly. Okay, used to find a specimen when using a low power objective. So when you're using the low power objective lens, which is the yellow one, we use the coarse focus knob. Okay, fine focus knob, this one, used to make small focus adjustment. Okay, so clarify the image, especially when using the high power objective. So when we use the high power objective lens, we use fine focus knob in order to clarify the image. The diaphragm, it's this one, the part that's found under the stage, rotating dial that controls the passage of light through the stage. Okay, it controls the amount of light that passes through the stage. Numbers of the dial indicate the relative amount of light passing, with 5 being the most and 1 being the least. Okay, it contains numbers. The highest number considered to be the most light passing, and the 1 is the least passing of the light. The base, this one, which is found at the bottom of the microscope, supports the microscope and contain the electronics. The microscope should be carried with one hand under the base and the other holding the arm. Light source, this one, the light source illuminates the specimen by shining bright light through it. Okay, which means to make it bright, because if the specimen is dark, we wouldn't have the ability to see it. The last part of the microscope is the power switch. The power switch turns the light on and off. That's it. Okay, this is its function. So these are the 12 parts of the microscope. Now we have the using of the microscope. So as we mentioned before, you see the picture how he held the microscope while carrying it from place to place. Carry the microscope with two hands, one on the arm of the microscope and one under the near the base. Okay, this is really important. Switch on the light from the light source or turn the mirror toward the light. Switch on this one in light microscope and this one in old microscope. They turn the mirror toward the light because there is no light source. Some materials viewed in dim light, it means in dark light, and other bright light. Light intensity is regulated by the diaphragm. The diaphragm, which is this part which is found under the stage, which said it controls the amount of light that passes through it. Okay, put the study material on the stage. Okay, we put it on the stage. Firstly, use the coarse adjustment to see something roughly. So the first thing we do when we want to see something in the microscope, we use the coarse adjustment because you're using a low power lens. And then you use the fine adjustment to see the materials clearly with the high power lens. Now we have some types of microscopes or magnifiers. The first type, or the simplest type, is the magnifying glasses. This one is very common and you know it and you have seen it. The oldest type is Koch's microscope. This one, which is the oldest microscope, on the first microscope. We have this one, which is the most common microscopes in labs. It's called the light microscope. And this one is the most modernist microscope and the best one, which is called the electron microscope. The electron microscope magnification wide from 10 times to 500,000 times. Okay, and 250 times the magnification of the light microscope. Okay, so it's magnify the sample 250 times more than the light microscope. Okay, so these are the types of microscopes. Preparing of object to be observed. The example that you have is an onion. An onion, we take a small part, cut a small piece of the onion, and then we take from this piece we peel with your fingernails a small tissue, very thin layer. We put it on the slide. This is the slide. Okay, you see it, a glass piece, transparent, so that allow the light pass through it. We put the piece of onion on it, the very thin piece of onion, and we add a water drop to on it, and then we take a cover slab. This cover is added over the sample, and then so that it will be clarify the image, and we get rid of the bubbles of air in the water. So, and then when we finish, we put it under the microscope. When we put it under microscope, we will see this image in the microscope. This image shows you see this parts. This represents the cells of onion. Okay, this one, each part is a cell of onion, contain a nucleus inside it, a cell wall, cytoplasm. Okay, so it's considered to be a normal cell, onion cell. Thank you.